In this video, we will discuss about in vivo study of RMC-6236 in immunodeficient and immunocompetent mice. So here what it is, they use this RMC-6263 first in the immunocompetent mice, that is BALB-C mice, and they inject the mice with this KRAS G12C mutant tumor cells, that is ECT26 cells. And once the tumors are injected, uh, once the tumors is injected, first they check it with a control where they are not able to see any MCR. When they check it with this only immune checkpoint inhibitor, it is anti-PD-1, they are only able to see 1 out of 10 MCR. Whereas when they used only RMC-6263, they are able to see 6 out of 10 MCR. So this result is much better than the immune checkpoint inhibitors alone. However, in the clinical practice, it is common to combine immune checkpoint inhibitor with the other drugs. So here they also did the same thing, that is they combined RMC-6263 with anti-PD-1 antibodies, and here they are able to see 10 out of 10 MCR. So in the immunocompetent mice, they are able to get around 10 out of 10 MCR. However, when they did the similar studies in the immunodeficient mice, that is NSG mice, which is also injected with the same tumor cells, that has KRAS G12C mutations, in that case they are not able to see any improvement, that is 0 out of 10 MCR, which is equal to the control. And then this shows that the immune memories is important in order for this RMC-6236 to function. Thereafter, they checked for this immunocompetent mice, they checked the presence of the CD4 and CD8 plus antibodies, which can act as a anti-tumor, which can function as an anti-tumor, and we are able to see their expression levels are significantly higher compared to the control, CD8 plus and CD4 plus, and these expressions are checked in the tumor microenvironment. So in the tumor microenvironment, immune system is very much active when they combine this RMC-6236 with anti-PD-1. Along with that, they also see the high expression of this MHCII, which is also anti-tumor factors, or which promotes the tumor suppressions. Afterwards, they also checked some of the factors that act as a tumor suppressors. These are they also check the other products that act as a tumor promoters. For example, these myeloid-derived suppressor cell factors such as Ly6C, Ly6G, this and M2 type macrophages, which also promotes the tumor proliferations. So all these three are also downregulated compared to the vehicle. This in altogether indicates that this RMC-6236, along when combined with these immune checkpoint inhibitors, it can not only leads to the lower MCR, but it will also activate the other immune systems and suppress the tumor promoter factors. And in the supplemental figures, they did the similar studies, that is immunocompetent mice, on this G12D ECT6, and there also they see the similar results, that they have the better performance when they combine RMC-6236 with anti-PD-1. So that indicates that, along with the results that are presented in this main figure, indicates that RMC-6236 is not only specific to the particular kind of KRAS mutations, but it can work efficiently on both this KRAS G12C and G12D mutations. So this is what they finally conclude from this figure. And one thing to note, that the amount of this RMC-6263 is used is around 25 mg per kg in all the cases, and this anti-PD-1 is used around 10 mg per kg. So, and these results are based on this dosage. So I hope you understood the results that are presented in this figure, and how they shows that RMC-6236 can work on multiple KRAS mutations. However, the results for the other KRAS mutations, that is G12D, is not in the main figure, but it is in the supplemental figures. So if you need to understand more about these experiments detailed and wants to refer to the supplemental results also, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.